Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. We would like to ask the questions how the information from the one generation to another generation is passing, and what are the different types of molecules which could be responsible for passing the information from the one generation to another generation. So now what you see is that I'm sure you might have noticed that some of your own traits are matching with your parents. Similarly, the traits what are present in the plants, they are also of mixed traits, right? They are also having the some information from the one parent and some information from the other parents. And on the other hand, you might have seen that some of the diseases which are propagating within a particular type of families. For example, I'm sure you might have noticed the traits like height, eye color, and other kinds of phenomena like the hair colors and the way you speak and the way you actually behave is all being transferred from the parents to the child. Similarly, you might have seen that the seeds from a red color flowering plant always produce red color flowers naturally, right? And then you might have seen also the different kinds of variations, like if you have the crossbreeding of a white flower and the red flower, you will see that they were actually having the pink flower and so on. And there is a classical examples of the hemophilia also, which actually runs in different types of families from generation to generation. Now the first question comes that how does it happens, and the answer to this question is that it is all because of the heredity. Okay. So what is heredity? So heredity, or also called as inheritance, is the passing on the traits from the parental generation to the offsprings, either through the asexual reproduction or the sexual reproduction. The offspring of the cells obtain the genetic information of their parents. Okay. So because of the hereditary or the inheritance, you are acquiring the traits, or you are acquiring the phenomena or acquiring the phenotypes from your parents. Definitely when if it is a asexual reproduction, the traits are going to be completely 100% intact. But if it is a sexual reproduction, then it is actually going to be mixed, because you are going to have the 50% traits from your mother and the 50% traits from your father actually. And depending upon which trait is dominating and which trait is recessive, it is actually going to show you the phenotype. So the first question comes that if it is actually the thing that you are actually going to acquire the traits from the mother and the father, how this particular type of phenomena is happening and who is responsible for that. So who is responsible for carrying the information from one generation to another generation? And the molecule which is responsible for this particular information or for this particular phenomena is called as the genome, or I will say genetic material, because it is actually going from one generation to another generation, and because of that this particular molecule is being called as the genetic material, not the genome actually. And I am sure when we were discussing about the cell, and even in the previous lecture also when we were talking about the biomolecule, if you see the cell is very complicated, right? It actually has a nucleus, it has the cytoplasm, it actually has different types of organelles. In the prokaryotic system also you have all those things, but not the membrane bound, right? So you also have the miniaturized level of, or the primitive conditions of the electron transport chains. You also have the other kinds of things. So basically if you think about the wherever the information can be stored, there are multiple possibilities. One is, in a cell it can be nucleus, or I will say the genome, or I will say DNA, because we know that the nucleus contains the genome, which is made up of the DNA. The second possibility is the cytosol, or I will say cytosol actually, right? and cytosol is mainly being contained one molecule which is called as protein and plus RNA, right? So RNA is also present in cytosol. Then you also have the membrane bound organelles. So membrane bound organelles are also being made up of the protein and lipids, right? Because the membrane and lipids, right? So we basically have the candidate molecules which are responsible for carrying the informations, but because the technique was not evolved, or how the people have find out the who is actually responsible for carrying the information is a very long journey, right? So let's first, so what are the molecules we have? We have the DNA, we have the potential molecules like the DNA, we have the proteins, we have the RNA, and these are the molecules which are actually having these stored informations. For example, you know that the DNA is made up of the nucleotides, so that also is actually providing the sequence of nucleotides, so that also can actually carry the enormous amount of informations. I'm sure you can calculate, if you have 4 nucleotides, how many different types of random combinations could be possible to give you the different types of random DNA sequences. Similarly, you have the proteins which are made up of the 20 amino acids, and all these 20 amino acids random combinations can give you the enormous information. So that's how, and the RNA also, right? The same way the RNA. So these 3 molecules are actually having the similar kind of nature. Apart from that, you also have the lipid, but lipid does not have that kind of flexibility of storing the information. So that's why the lipid is straightforward, is being discarded by the scientists. What they were focusing on the DNA, protein and RNA. But before that, the people have done the crude experiment, because until the people have not discovered the DNA, protein and RNA, they were not having the technology. So they were doing the experiment, they were trying to identify the carrier molecules which actually carries the information from one generation to another generation, and that's how they have done multiple type of experiment. So let's see how we have, what is the history of identifying the genetic material, and then we will going to discuss about the different types of classical experiments, how people have figure out whether the DNA is the genetic material or protein or RNA. So the first experiment is been done by the Austria scientist, or I will say the priest, Gregor John Mendel. Okay, so he is considered to be the father of genetics, and he actually has done extensive experiment with the pea plants, and where he has taken a combination of traits, and that's how he have come up with the classical rules of the genetics. And so what is the history of the genetic material? Okay, so it is well known that the qualities are passed down from one generation to the next. The offspring shares certain characteristics with both of their parents, but the question is, who is responsible for this? It was first time, on the basis of its interest in the plant hybridization studies on the sweet pea, the Pisum sativum, monk in a monastery in Austria, Gregor John Mendel, tried to find out this answer. He proposed that the some factors, so in when the Gregor John Mendel was doing the experiment, there was no technology evolvement, there were no technique available to say that DNA, RNA or protein, but he said that there are factors, and he used the term factors, that carry the information on the manifestation of a characteristic or phenotype, and how the traits are being passed from one generation to another generation. Since he gave the clue about this particular factor, and he said that this factor could be dominant factor and recessive factors and so on and all that, and all these law of genetics you might have studied in some of the textbooks. So then people have started identifying this particular factor. So in the late 90s century, by the 3 biologists, Hugo de Vries, Carl Cornes and the Eric Vaughan, worked on the Mendel works and proposed that different characters have individual hereditary carriers and the inheritance of specific traits in an organism called particles. So De Vries actually called these units as the pangenes. So, and he actually come up with the some of the theory also that so he actually discovered that these factors which the Mendel was talking about is nothing but it is actually the pangenes, which actually and if the pangenes can move from one generation to another generation, they are actually going to carry that particular characteristic or the quality. But in the almost 20 years later, when the William Johnson and William Watson actually proposed the term gene and the genetics respectively, but Edward Strasburger and the other continue to refer to the basic physical and functional unit of heredity as the pangene. So basically from the pangene it becomes gene, and the gene was being considered to be a responsible factor, right? So genes are present in chromosome which are evenly distributed between the two daughter cells during the cell division, and the biochemical study showed that the chromosomes are consist of protein and the DNA. So it is clear that the gene is present in chromosome, right? And this we are going to discuss about this nuclear packing and all that when we are going to talk about the genetic material. And the chromosome is made up of two parts, right? The protein part or the DNA part. So the first question is that is the genetic material protein or the DNA, which means out of this chromosome which one is more responsible, the protein part or the DNA part, because it has both the components, right? So until 1940s, the proteins were thought of a genetic material because proteins are polymer made up of the 20 different types of amino acids which are abundant and encode the diverse informations. And you can easily calculate. In fact, you can actually go with that activity, right? How many different types of amino acid compositions or amino acid combination could be possible if you have the 20 different types of amino acids. For example, you have a very small protein of 100 amino acid. If you have a small protein of 100 amino acid and you can have the 20 different random combinations, you can actually be able to calculate that number. That number is going to be very, very, very big actually. However, based on the certain experiments that have been conducted from time to time, it was finally shown that the DNA, not the protein, actually carry the genetic material. But we are actually going to discuss in detail about these experiments. And the conclusion comes that it's a DNA which is actually being the molecule present within the chromosome, and that is actually going to carry the information from one generation to another generation. Now the first question comes, what could be the properties of the genetic material? What could be the possible or the probable properties you should have, or a molecule should have, then only you can say, okay, this is the genetic material. So the properties of the genetic material, okay. So for a molecule to be considered as the genetic material, because you can have some requisite parameters to accomplish its task, right? What is the task? Task is to carry the information from one generation to next generation, right? That is the task of a genetic material, and that's why it should have a prerequisite conditions. What are the conditions? Number one is stability. Number two is it should be heritable and expression. Number three, it should have the mutations, and number four, it should be getting replicated. So stability, it must contain all the biologically useful informations in a stable form, which means it should be stable, it should be it should be resistance for any kind of damages, and if there is a damage, it should have a mechanism to recover from that particular damages. Then it should be having the heritability and the expression. So it should process hereditary units which follow the Mendelian inheritance and control the expression of a particular phenotype. So it should have the components, it should have the, and I think all these you are going to understand when we are going to talk about the transcription and translation, that how there are different components which are present within the genetic material, and that actually controls the expression of a particular gene. And So you should have these kind of switches. You can have these kind of switches so that you can actually be able to modulate the expression of the particular gene, and that's how you are actually going to modulate the overall phenotype of that particular organisms. I'm sure you might have noticed when you are going into the sun, right, and it is very hot outside, you always start sweating, right? So that sweating occurs because there is a expression profiling changes within your body, and that's how they started throwing the sweat. Same is true when you are entering into a into a cold room, or entering into a place where you have AC, and then you actually all your sweat is disappeared, and then you also feel cold, right? And when you feel cold, you find that the skin is actually becoming more contract actually. Okay, so these things actually been done simply by the information what is present inside the genetic material. Then the third property is about the mutations, right? So genetic material is also going to acquire the mutations. Some of the mutations could be good, some of the mutations could be wrong, and that's how the accumulation of mutation is actually going to result into change in the phenotype, and that will be responsible for evolution, right? So mutation is the random change which may occur, and that may be a chance for evolution actually. And then number 4 is replication, because as I said, genetic material is, you're going to have only one copy of genetic material, so that genetic material has to be replicated, and that's why you're going to have 2 copies of genetic material, and then you can actually be able to, in a situation that you can be able to share. Remember that when we were talking about the cell cycle, we said that, right, during the S phase, the DNA is actually going to be duplicated, right, and then only it is actually going to be shared between the cell. And the same is true for between the parents and the offspring as well. That you're going to have the 2 copy of genome, and then when you are going to share 1 copy with your offspring, and the so on. So that should have the ability to synthesize its own copies. So this replication is actually what we are going to discuss in our subsequent module. We are going to talk about the replication, transcription and translation. Now there are some direct evidences or experimental evidences, and there are some indirect evidences which prove that the DNA as a genetic material. Okay, so there are direct as well as indirect evidences which have ruled out the protein or the RNA as a genetic material, and that has proved that the DNA is actually the most acceptable genetic material which is present starting from the prokaryotes to mammals. There are exceptions, and these exceptions also we are going to discuss. So in 1928, Frederick Griffith actually performed an experiment for the bacterial transformation, and by doing these experiments he proved that actually the information what is being present in the DNA is actually carried from one bacteria to another bacteria, and that's how it is actually going to change its phenotype, and that's how it is actually going to be responsible for the death of the mice. Then in 1944, the Oswald, Avery and McLeod and McCarthy actually done the experiment on the transformations, and that also has proved that it is actually going to be the DNA which is going to be responsible factor. And then we have the Alfred Hershey and the Chase experiment on the T-even bacteriophage, and that's how that also has proved that the DNA is a genetic material. So if you go by the timeline, in 1869 the Frederick Metcher actually isolated the nucleic acid, or I will say the genetic material, from a white blood cells. Then in 1928, so almost after 50 years or 60 years, the Frederick Griffith actually demonstrated that the genetic information from the one bacteria goes into another bacteria through a phenomena which is called as the transformations. Then in the year of 1944, the McLeod and McCarthy actually identified that the DNA is actually a transforming agent. It actually carries the genetic information, and then it actually can change the phenotype of the other bacteria. Then in the case of 1952, your Hershey and Chase actually confirmed that the DNA is a genetic material with the help of the viruses. And then in 1962, the Watson, Crick and all those people have got the Nobel Prize, because when they have discovered the structure of the DNA. So let's first discuss about the Griffiths experiment to understand that the DNA is the genetic material, and then we will talk about the McLeod and McCarthy's experiment, and then at the end we are going to discuss about the Hershey and Chase experiments. So the Griffiths experiment, so he has used a bacteria which is called as pneumococcus. He has used the bacteria which is called as Streptococcus pneumonia, and he has used the two different types of strain, S strain and the R strain. The S strain, which is a virulent pathogenic strain, that is S strain because it's called a smooth strain or the S third strain. And the R strain is a recessive strain. So R strain is the avirulent strain, and it is non-pathogenic strain known as the R virulent or the rough strain or the R2. So S strain is actually going to cause the disease, whereas R is not going to cause the disease. So if the S is going to cause the disease, S is actually going to kill the mice, whereas R is not going to kill the mice. So S strain has a smooth outer coat made up of the polysaccharides, and the R strain lacks this polysaccharide coat, and therefore its surface appears rough. S3 strain was virulent, possessed a lipopolysaccharide capsule and could kill mice by causing the disease pneumonia, and made round colonies on a cultural plate. Whereas the R2 strain was avirulent and lacked a lipopolysaccharide capsule, giving rise to the rough shaped colonies onto the cultural plate. So these are some of the properties given, right? So stereotypically it can be R2 or the S3, and morphologically the R strain is a rough strain, whereas the S strain is a smooth strain. Then capsule was absent in the case of R strain, whereas it is present in the case of S strain. And the R strain is avirulent. What is mean by avirulent is that it is not going to cause the disease. Whereas in the case of S strain it is virulent, so it is actually going to cause the disease. And what disease it is going to cause? It is going to cause the pneumonia, and ultimately it is actually going to cause the death of mice. So this is the experiment what he has done. So he has done the experiment using the 4 different types of conditions. So case 1, case 2, case 3, case 4. So in the case 1, what he has done is he has taken the S type of bacteria, and that he has injected into a healthy mice, and after some time the healthy mice is gone, right, because he developed the disease, right, pneumonia. He developed the pneumonia, and that's how he actually died. In the case 2, he has injected the R type of bacteria. Okay, so R type of bacteria is avirulent bacteria, so it is actually not going to cause any kind of disease, and that's how this particular mice is actually going to grow, and that's how the mice is actually going to live. Then the third experiment what he has done is he has actually denatured the S type of bacteria. So he has heat killed the S type of bacteria, and then he injected that into a healthy mice, and that also has not caused any disease, and that's how the mice is survived. Then the third is he has taken a heat killed S type of bacteria. So virulent bacteria he has heat killed. So this is not live bacteria. And then he added the R type bacteria which is live. So this is live, this is dead bacteria. And then he mixed them together, and in the case 4, and then he injected them into a healthy mice. And when he had done that, he actually found that the mice have developed the disease, and they also died. So this was very interesting, that something which was not infective, because in the case 2 you see that the R strain is not killing, but in the case of case 4 the R strain is killing the mice if it is being mixed with the heat killed S3 actually. So these are the 4 different types of conditions, right? And then what he has done is he has isolated the blood, right? He isolated the blood from this case number 4, and what he found that there was no R bacteria present. It was all the S type of bacteria what is being isolated. So all the R type of bacteria is actually being converted into the S type bacteria in the case 4. And that's how he said that the material or the material which actually carries the information from this bacteria, material gone from the heat killed bacteria into a R type bacteria, and that's how the R type bacteria got converted into S type bacteria. So there is a, and then that's how he actually coined the term that this phenomena is going to be called as transformations. So when the Griffith has injected a mixture of heat killed and live bacteria, the mice died and living S bacteria recovered from the dead mice. Okay, so remember that he has actually injected a heat killed S type bacteria, but he got the live S type bacteria because the genetic material what was present in the S type bacteria, heat killed bacteria, were gone into the R type bacteria, and that has that has actually started expressing its own genome, and that's how it is actually going to cause the generation of S type bacteria. So Griffith concluded that the R type bacteria had somehow transformed by the heat killed strain bacteria. Some transforming principle transferred from the heat killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become the S strain. This must be due to the transfer of the genetic material from the S type bacteria to the R type bacteria. However, the biochemical nature of genetic material was not defined from this particular type of experiment, right? So we don't know still that what is actually being transferred, whether the DNA or protein or RNA, right? So then they did the further more experiments. So in the meantime, the Oswald and McCarthy and McLeod actually did the more experiment, more specific experiment to ask whether the factors which are actually converted the R type bacteria into a S type bacteria, whether it is the DNA, protein or RNA. So it was believed that the genetic material was made up of the protein. They worked on the transforming principle in Griffith experiment to investigate its biochemical makeup. So to determine the biochemicals from the heat killed S type bacteria which could convert the live R type bacteria into S cell, they isolated the biochemicals, like they isolated the proteins, DNA and RNA from the S cell bacteria, and finally found that the DNA from the S bacteria was needed to convert the R type bacteria. So what they have done and what they have used for this particular type of experiment, they have used 3 different types of enzymes. They have used a DNases. So the function of DNA is that it is actually going to digest the DNA. So it is actually going to destroy DNA. So if you take the DNase and if you treat any reactions, it is actually going to destroy the DNA. So it is actually going to remove the DNA part. Similarly, they have the RNase. So RNase, the function is that it is actually going to digest the DNA, which means it is actually going to destroy RNA part, or I will say it is actually going to remove the RNA from the reactions. Similarly, you have the another enzyme which is called as proteases, and the target substrate for the proteases is that it's actually going to digest the proteins, and it is actually going to destroy the proteins because it is actually going to convert the protein into the amino acids. This means it is actually going to remove the protein components. This means if you have a reaction, right, for example, if you have a reaction and if you treat this with the protease, for example, right, so then this reaction is going to be a reaction minus protein because you have treated with the protease, and that's how you have removed the protein. So these are some handy enzymes what these people have used to answer the questions which biomolecule is actually the genetic material. So what they have done, they have repeated the same experiment what the Griffith has done, that where you have the heat killed S type bacteria and then you have the live R2 bacteria, and then when they were taking the heat killed bacteria, they have treated that heat killed bacteria with protease, right. So they have either treated it with the protease or treated it with the RNase or treated with DNase. Okay. So in these cases, what you have done is you have removed the protein, right. In this reaction, you have removed the protein, in this reaction you have removed the RNA, and in this reaction you remove the DNA, right. This means in this reaction you have the protein and RNA present, in this reaction you have the DNA and protein present. So what are the things present? You have the DNA and RNA present, right. You have, because the protein is already been removed because of you have treated with protease. Then in this one, since you are removing the RNA, you are going to have the DNA and protein, and in this one you are going to have the RNA and the protein because you have removed the DNA, right. And then he tested in which condition the mouse is actually going to die. So in this case, when you have no protein, mice is dying, right. This means the protein is not responsible for converting the R bacteria into a virulent S bacteria, right. Similarly, when you have no RNA, right, then also the bacteria mice is dying because still the R type bacteria can be getting transformed into a S type bacteria. And the third is when you don't have DNA, right. So when you remove the DNA, you are actually going to see that you're having a healthy mice, right. This means in this one you have the RNA and protein, but still the mouse is not getting the disease, which means the R strain is not getting converted into S strain, and that's how the mouse is actually healthy. And what you see even at the cultural level also, when they cultured the bacteria, what they found that they could be able to recover the live S bacteria in the absence of protein and RNA, but they could not be able to isolate the live S bacteria. Instead, they are actually getting the R type bacteria, R2, in the case of the third condition when their DNA is not present. So this actually confirmatorily proved that DNA is the genetic material. Now, let's talk about the conclusions. So, they found that both RNA and protein digesting enzyme had no effect onto the transformation, proving that the molecule undergoing transformation were either protein or RNA, okay, or neither a protein or RNA. Transformation was prevented by DNA digestion, indicating that the DNA was the transformation agent. So, they came to the conclusion that the DNA is the genetic material, but not all biologists agreed with that, okay. And then the further experiment were done by the Alfred Hershey's and Hershey and Chase experiment. So in the Hershey-Chase experiment, the researchers on the virus that infect the E. coli provided additional evidence for the genetic importance of the DNA. The DNA core of the T2 bacteriophage is encased in a protein code. Alfred Hershey and Hershey Chase put this theory to the test in the following manner. The radioactively labeled T2 phase in either the protein, which means the 35S, or they have labeled the DNA with the help of the 32P component before injecting them into the bacteria, which means they are going to have the bacteriophage where they have labeled the protein or where they have labeled the DNA by the radioactivity. Specifically, they infected the non-radioactive E. coli with the radio-labeled T2 bacteriophage. So, in the T2 bacteriophage, you can have the protein which is 35S, which means sulfur labeled, or you can have the DNA which is actually going to be the 32P, right, or the phosphate labeled, okay. And that's how they have asked where or which molecule is going from one generation to another generations. So, they injected the bacteria with the T2 phase that has been radio labeled either in the DNA component or in a protein component. The infected bacteria were agitated in a blender and the two fractions were separated by the centrifugations. One fraction contains the empty fast coat that was released from the surface of the bacteria that consists of the protein and therefore carries the 35S radio label. The other fraction consists of the infected bacteria itself. Most of the 32p level was found into the infected bacteria, which means when they were doing the agitation, what they found is that the protein which is a part of the coat was always being extracellular and it was not being carried within the bacteria, right? It was not getting into the bacteria. And so when the virus is infecting, the its coat is remain outside, and that's how it is actually going to be present outside. This means the radio-labeled what radio-labeled protein remain outside, whereas radio-labeled DNA remain inside. So that's why there was no sulfur what has been associated with the bacterial cell, whereas in the case of DNA, all the DNA was present inside, and that's why there, after centrifugations, the radioactivity was associated with the bacteria. So by doing this experiment where they were doing the infection followed by blending and followed by centrifugations, what they found is that the protein is extracellular, whereas the DNA was associated with the bacterial system. And by doing this experiment, the Hershey and Chase concluded that the DNA is the genetic material. So by the observation, they found that the most radioactive protein was released into the supernatant, whereas 32p DNA remained within the bacteria. Since genetic material was injected and T2 progeny was produced, DNA must have been carrying the genetic information for the T2. This means not only that, when the bacteria got lysed, it has produced enormous amount of viruses. This means the genetic information was there inside the bacteria to produce the viruses, and that was nothing but the DNA, right? As ghost or the coat of the bacteriophages were not labeled with the 32p and only with 32s, the result of the experiment clearly indicate that the only DNA and not the proteins enter the bacterial cell. Protein coat is left outside. All of the genetic data necessary for the creation of a new phage particle is carried by the DNA that entered the host cell. This undoubtedly demonstrated that the DNA, not the protein, serves as the bacteriophage genetic material, and that's how they have concluded that the genetic material is DNA, not the protein. Okay. Then there are several indirect evidences for DNA to be as genetic material. First evidence is that the DNA regularly present in the nuclei of all cell types. It is equal the amount of DNA present in all cells of an organisms. Then amount of DNA is proportional to the ploidy of the cell. Haploid cells have the half amount of DNA than the diploid cells. Nuclear division occurs only after the DNA duplications during the S phase of the interphase. This anyway we have discussed when we were discussing about the cell division. Then the different species have different amount of diploid DNA. Out of all macromolecule, DNA is metabolically more stable or the most stable molecule, and that is the first criteria, that the genetic material should be very stable. Indefinite number of combinations are possible with the 4 sub-bases like ATGC. DNA has some same physical and chemical property in all organisms, yet allowed to produce great diversity of the organisms. So this was clear that the DNA is the genetic material, right? And material. But this has been challenged when people have discovered that there is a phenomena which is called as the reverse transcriptase or reverse transcription. When the people have discovered a phenomena which is called as reverse transcription. So what is mean by the reverse transcription is that the RNA is actually going to give rise to the DNA. Okay. So this reverse transcription was against the central dogma of molecular biology, right? It says that the RNA can be able to produce the DNA, and by doing so, there was people who said that RNA is also made up of, it's very stable, right? RNA is also stable. RNA is also can provide the diversity, and since RNA can be converted into DNA, is there is a possibility that the RNA could also behave like a genetic material, right? And then the same in under the exceptional cases or some other kinds of cases, right? So then again the same debate started whether the RNA could be a genetic material or not. So to prove that, the people have started doing the experiment. Okay. So RNA as a genetic material. Okay. So according to the RNA world hypothesis, the RNA was the first genetic material that stored all genetic information, and it is believed that the first life arose from it. RNA is thought to catalyze a number of chemical reactions in the primitive cell. The presence of the 2-hydroxyl group in ribose group increase their reactivity, but this reactivity makes them unstable, which makes the RNA unfavorable as a genetic material. As genetic material, it should be well stable chemically and structurally. So that was one of the drawback of RNA as a genetic material, that it actually contains the 2' hydroxyl group, and because of that it is actually having the more reactivity compared to DNA. So ultimately these unstable molecules are replaced by the more stable genetic molecules. During this stage of evolution, the DNA molecule emerged. They have replaced RNA's role as both genetic material and structural component. The unstable and degraded nature of RNA has led to the development of double stranded DNA genetic material that is both chemically and structurally more stable. So according to the hypothesis, it is found that the RNA is actually being the preferred material for genetic material in the primitive cell. Okay, so during the evolution, what the cell has found, that RNA is good in terms of genetic material because it reduces the steps, right? You don't have to go for the transcription, you can directly use that particular information to produce the protein. But on the other hand, it is unstable. So what they have done is they have converted the RNA into a double stranded DNA, and that's how you are actually bringing the more stability and chemically as well as structural stability into the structure. However, RNA is not being completely eliminated. They still serve as a genetic material in some systems like the viruses, and they catalyze few essential biochemical reactions into the cell. Also, the complex machinery of protein synthesis from DNA is still proceeding through RNA. Okay, so RNA is very important in terms of relaying the information from the DNA, and that's why it is not been excluded from the complete picture of the protein synthesis. Still the protein is being synthesized from the RNA itself. So even if you see that the DNA actually stored the information, it is RNA who actually dictate the production of the protein, and that's how RNA is actually responsible for the particular type of phenotype. So RNA is present as a genetic material in some of the viruses, right? So virus consists of two parts, nucleic acid and the protein coat, sometimes with additional envelopes. So virus contains only one type of nucleic acid, either the DNA or the RNA. Viruses have RNA are called riboviruses. They vary in the structure of their nucleic acid. Most of the plant viruses are RNA viruses, either single stranded or the double stranded. So you can have the animal viruses, you can have plant viruses, you can have single stranded RNA viruses, you can have double stranded RNA viruses, and even in the plant viruses also you can have the single stranded viruses like the TMV, or you can have double stranded viruses like the Oriya viruses. Then we have the evidence in favor of RNA as a genetic material. The first evidence that RNA is also has a capacity to carry the genetic information came from the experiment which is conducted on the tobacco mosaic or TMV viruses. So Krieger and Schumann demonstrated in 1956 that the tobacco plant can contract mosaic disease when exposed directly to pure RNA from TMV. RNA's pretreatment rendered the pure RNA incapable of inducing the TMV lesions. Then Franklin, Conhart and Singer demonstrated in 1957 that the progeny viruses from the TMV infection with viruses have RNA from one strain and protein from another strain, but invariably of kind determined by the RNA, not the protein. So this is what exactly they have done. They have taken a TMV virus, and what they have done is they have removed the capsid protein. So they have removed it, fragmented that into a protein and RNA, and then they degraded the RNA with the help of the RNase. So these are the four components. So when they take the TMV virus and if they infect the new cells, new leaf, they could found that the infection is happening. When they remove the capsid, okay, when they remove the capsid part, right? So still it is actually having the RNA, right? So if they remove the fractionated that into a capsid and the RNA, when they taken the capsid protein and infected that to the protein, there is no infection. So there is no infection, right? No infection. But if take the RNA and if that infected into the protein into the leaves, what they found is there is an infection. So basically, the genetic information what was present into the RNA is good enough to produce the virus, and that's how it actually causes the disease. When they degraded the RNA with the help of the enzyme RNase, so there is no RNA, right? They could found that there is no infection. So there is no infection in this case also. So Grigor and Schumann correctly concluded that the viral genome of TMV is composed of RNA. So if there is no RNA, then there will be no infection. And then Franklin, Guwahati and Single uses the type A and type B TMV virus in this investigation. The RNAs were isolated from the protein coating, and then in order to create hybrid viruses, Singer concluded the RNA of the strain with the protein of the another. So the phenotypically and genotypically identical progeny virus was similar to the parental type from which the RNA had been recovered after rubbing the hybridization or reconstituted viruses onto the living one. So these people, what they have done is they have excluded the RNA of, they have isolated the RNA of from the two different types of TMV viruses like TMV-A and TMV-B, and when they mix them together, what they could found that they are actually having the the hybrid viruses what is being produced. So in conclusions, Franklin, Guwahati therefore come to the conclusion that both the DNA and RNA can carry the genetic information. As a result of all these investigations, his research established that the genetic material for TMV is stored in RNA rather than protein, and however, the DNA may always serve as a genetic material, but RNA is typically non-genetic. RNA only serves as a genetic material in few instances when DNA is not present. So in summary, what we have discussed that the gene carries the data for the phenotypic expression, and these genes are being called as the factor in the case of Mendel. And the chromosome have the gene on them. They are made up of the 60% protein and 40% DNA, and stable genetic material that can replicate, store information for expression and undergo the mutation is required. And experiments by the Griffith, Avery, Hershey and Chase have produced results that directly supports the DNA as the genetic material. There are also some circumstantial arguments in favor of the DNA as the genetic material. So experiment on TMV by the Grigor and Schumann demonstrated in 1956 that it is the RNA which is actually carrying the information from one generation to another generation, and that's how wherever you have the RNA, it is actually going to be responsible for generation of or causing the disease. Even when they have mixed the two different types of viruses, what they found is that they are actually generating the hybrid viruses, and the protein has no role in carrying the information from one generation to another generation. So by doing all these experiments, it is concluded that the RNA is not being preferred as the genetic material until the DNA is present. But if the DNA is absent, the RNA is also being taken up as a substitute for carrying the genetic information from one generation to another generations. Or I will say that RNA is being preferred by the primitive organisms or such as viruses, and whereas DNA is, because DNA is more evolved, so DNA is being taken up by the higher organisms. And that's why very categorically you can see that RNA is one of the most preferred genome in the case of the viruses, whereas the DNA is more preferred in the case of higher animals. So, with this brief discussion about the genetic material, we would like to conclude our lecture here. In our subsequent lecture, we are going to discuss some more properties of the genetic material and how it actually has a role in synthesizing the protein, and we are also going to discuss about the central dogma of molecular biology. So, with this, I would like to conclude my lecture. Thank you.